Okay, Doctor.
All right. Okay. Thank you for the signal. Right. I actually gave this topic to Chet Alha last time when he asked me what I would like to share with you this morning. So it's very general because since my research life started during my master's back at UTM in 2004, starting in 2004. So I've done a bit of everything, spanning from microbiology, biochemistry, and molecular biology. So I regard that as, me doing try and error, trying to explore what is my research interest. And you can see hopefully from the presentation in a bit how my previous experience actually shaped what I'm doing right now. My research journey from UTM until my PhD and all the way to my postdoctoral attachment at NUS, which has just been completed. So, overall, I can say that my research interest is slowly, getting in shape. And I would say that this general topic, microorganisms for the environmental and industrial applications, actually summarize my current interests quite well. Okay, so before I go any further, I just would like to share with you, even though our chairperson has briefly shared with you about myself, so a little bit more to be shared with you. So I am actually attached to Faculty of Science, UTM, as a senior lecturer at the moment, and I've just completed my postdoctoral attachment at NUS, particularly at the Environmental Research Institute. And I am also affiliated with the Synthetic Biology for Clinical and Technological Innovation. They call it SynCity at NUS. And I'm also the chair of the American Chemical Society, or ACS, Malaysia chapter. I completed my PhD education at Imperial College back in 2012, and I did my MSc and BSc both at UTM. So I have some professional affiliations listed here, and my current research interest is actually in the field of environmental biochemistry and microbiomes. And other than academic research, I am also passionate on chemical safety and security education, science communication and advocacy, STEM education, and also science diplomacy. So back when I was doing my MSc, I was actually involved in treatment of textile wastewater using biological methods. So my research group at that time, led by Professor Noraini and also Dr. Chan Gyakfa. So we were actually researching methods using microorganisms to treat colored wastewater. The reason why this research was being done at UTM is that in one area in Batu Pahat in Johor, there is quite a high concentration of textile industry. So they were trying to find a way to treat their highly colored wastewater, and biological methods was one of the methods being considered at that time. So what we did was to go out and find microorganisms in the environment that we can use to treat this colored wastewater. So my MSc research actually culminated in this publication where I use a microorganism called Citrobacter frundii to look at its capability to degrade different types of azodyes, as you can see here, and particularly in the lab. So since I was more interested in the biochemistry of the degradation, so my research was actually to characterize this particular microbe in the lab from its enzymatic activity, particularly flavin reductase activity, and what are the cofactors required by this enzyme. So in this case, you can see I was looking at the effects of NADH and riboflavin on the enzyme activity, and we came up with this particular scheme to show that this enzyme flavin reductase in Citrobacter frundi actually require cofactors NADH and riboflavin to transfer the electron to azodide in order to reduce it, to cut the azo bond, giving aromatic amines, and these aromatic amines can then be further degraded by other microorganisms. Other than that, I was also looking at the different redox potential of the different azodides, and I tried to correlate the redox potential with the reduction potential and also the extent of degradation by the enzyme flavin reductase. So we can see that the higher the redox potential of this azodide, the harder it is for the enzyme or for the microbe to degrade it. And at the same time, our group was also doing further investigation into this particular strain, Citrobacter sp, the same organism that I was working with. And around the same time as my MSc paper publication, we published the genome sequence of this strain. And with this information, we have a lot more knowledge about this particular strain. What does it do? What are the genes involved? And also what are the functional pathways undertaken by this strain during the degradation? And when I was doing a bit more reading on, when I came back from my PhD, I was researching on the topic that I would like to do for my research post-PhD. And when I was doing some reading of the literatures, I found that Citrobacter actually have been used and have been reported by many groups as one of the strains that we can use to produce biohydrogen. So I thought we already have the strain in the lab, and it can be used to produce hydrogen from waste materials, particularly. So I thought, let's try to investigate the ability of this strain that we have using azodide as substrate, and see whether it can convert it to biohydrogen. So why hydrogen? So compared to fossil fuels, if we have hydrogen as energy carrier, if we look at the combustion equation here, you do not produce CO2, only water. So even currently, hydrogen is actively being researched as fuel carrier for many different types of applications, such as transportation, electricity generation, and so on. So I thought this is interesting if we can generate hydrogen in a biological way, because currently most hydrogen being used in the many different types of applications are still from fossil fuel sources, as you can see here, and only about 4 to 5% coming from cleaner source, which is water electrolysis. So I thought biological hydrogen production is very attractive, because if we have waste materials, we can actually undertake this dark fermentation process by feeding the microorganisms any different types of waste that we have, whether solid or liquid, and generate hydrogen from there. At the same time, treat the waste material. So that is interesting for me. I thought that's a good idea for research. Unfortunately, our preliminary study, we found that azodide or Citrobacter frundi could not actually produce, even though it can degrade azodide, it does not produce hydrogen from its metabolism. So I thought then I must find a different substrate in order for me to investigate this biological hydrogen production. And again, in the context of Johor, we have a few industries dotted around Batu Pahat, Layang-Layang area, the cassava industry. So when I went to some cassava processing facility, I found that during the processing, cleaning, cutting of the cassava, they just release the water that they use into the drain. And the wastewater generated is actually rich in starch substrate from cassava. And I thought this can be another potential source of substrate for biohydrogen generation by microbes. And this is another research that I initiated in the lab. And so you can see the overall research pathway here. Starting with characterization of the starch wastewater using different chemical methods. And then initially we use a culture-dependent method. If you are familiar with microbiology, you know the difference between culture-dependent and culture-independent. At first, we took the culture-dependent approach by isolating the microbe from cassava waste environment, and then we ferment it in a strainer bottle like this, and then detect if there is any hydrogen being produced by the microbe that was being isolated. And then, normal microbiology method to identify them in 16S rRNA, and we look at the phylogenetic analysis. And also we did some kinetic analysis of the hydrogen production. And finally, what we did was to develop a consortium of starch degrader and biohydrogen producer, since there was a possibility that the efficient biohydrogen producer are not the most efficient starch degrader. So consortia is one way to do it. And fortunately, we managed to find a few strains that could degrade starch, as you can see here from plate assay. So these are the different strains that we isolated that can degrade starch and also produce hydrogen. Some can degrade starch but could not produce hydrogen. Some can produce hydrogen but could not degrade starch, something like that. So we did some biochemical analysis, looking at the amylase activity along the fermentation route, looking at the starch concentration, the biomass, which is the growth of the bacteria, and also the glucose concentration, which is products of starch hydrolysis by amylase. And we also look at the trend of hydrogen production at different physicochemical parameter. So as a conclusion, we managed to produce hydrogen using starch as waste materials with different strain as biocatalyst. But the challenge with starch is that you need to pre-treat it first. You cannot simply feed the raw wastewater directly to the microbes, because starch is present in granules, and in order for the microbe to access the granules, we need to pre-treat it first using a heat. That means we must hydrolyze it first so that it becomes readily available for the microbe to use and convert it to hydrogen. So I thought this hydrolysis process is actually energy intensive, and the overall process might not be very attractive to the industry if they have to first treat the starch using energy and then after that ferment it to produce hydrogen. So probably the total energy production is not attractive enough for the industry to take up the project. And then I switched to a different abundant substrate. I think most of you know that we in Malaysia have a lot of palm oil industry, and one of the highly generated waste is the palm oil mill effluent, the liquid waste. So we call it POME. If you go to any palm oil mill, you can see that they have ponds upon ponds of POME being treated using ponding method. And it would be interesting to find an alternative treatment strategy, or if we can treat it at the same time, produce energy for the mill to use back, it will be attractive for the industry to take up. So the next substrate that I tried was POME, and it is a two-pronged strategy, which is to treat POME at the same time produce energy in the form of hydrogen. So this is the research strategy that we took. First, to characterize the POME as substrate, and the microbe that we use this time is from anaerobic sludge. Since the culture-independent method nowadays are getting more and more improved, meaning we do not have to do isolation anymore. And sometimes when we isolate a single strain, when we give them raw substrate like this, they do not like it. So we stick with culture-independent method where we do not isolate the strain anymore. We just use anaerobic sludge directly from POME sources, and the microbe is already in there. So there is a mix of different native microbes in there that we can use to degrade POME and at the same time produce hydrogen. And the culture-independent characterization of different environments are also getting better nowadays. So we also took up the method 16S rRNA amplicon and also metagenomic sequencing to characterize the microbes present in the anaerobic sludge. So who are there to help us degrade POME, and then once we have some idea of who are there, what are the communities, we ferment the POME with the sludge in a reactor called CSTR. So it's a continuously stirred tank reactor. And the next thing was to monitor hydrogen production and also the carbohydrate consumption, VSS, which is the volatile suspended solid in the reactor, the COD, the chemical oxygen demand, and also microbial community changes, if there's any difference in the microbial community during the fermentation process. And the next thing what we did was to optimize the physical-chemical parameter of the process using a technique called the design of experiment based on RSM or CCD. So it's a software-based experimental optimization method, and that can be tested and replicated in the lab using experimental approach. So another experiment that we are doing in parallel with the fermentation here is to do what we call bioelectrochemical system. So bioelectrochemical system can be divided into microbial fuel cell and microbial electrolysis cell. So in general, if you're not familiar with this method, we use microorganisms. So it is like electrochemical cells where you have anodic chamber, cathodic chamber. So we add substrate on one side together with a specific electrode in the presence of the microbes. So the microbes can actually, while degrading or using up substrates in there, generate electron that can be used to produce electricity. So that is what happens in MFC. And in MFC, by tweaking the system a little bit, we can actually produce hydrogen in the fuel, in the cell. So MFC and MEC are more or less similar, but MFC produce electricity and MEC can be used to produce hydrogen, both from degradation of organic substrate by the microbes. So at the same time, we are also characterizing the how the microbe behave in the chamber based on the biofilm development at the electrode, and at the same time monitoring the performance. And then we optimize and also monitor what is happening at the electrode using microscopy technique called CLSM, also electron microscopy, and analyze the community using omic sequencing. And our ultimate goal is to integrate the fermentation methods with bioelectrochemical system in order to maximize POME degradation and also harvest more energy from the substrate in the form of hydrogen and also electricity. So I will just share with you some results from our recently published paper. So most of the work that I'm gonna share with you is actually done by my PhD student, Jamila. So what we did was to acclimatize the anaerobic sludge to increasing concentration of POME first, because we are afraid that using 100% POME concentration might be a bit of a shock for the microbial community and the anaerobic sludge. So we slowly acclimatize it to lower POME concentration, monitor hydrogen production at the same time, and then increase the POME concentration to 30%, 60, 100% in the end. So the whole process took about 40 to 50 days. And at the end of these 50 days, we get a community in the anaerobic sludge that are acclimatized to high POME concentration, at the same time can produce hydrogen in the reactor. The first analysis that we did was to look at the community changes during the acclimatization process. And you can see here the different relative abundance based on phylum, class, and families. So looking at the phyla level here, we can see that after acclimatization to 100% POME, this is what the TRT 100P means. So from the more diverse community in the untreated sludge, we can see that slowly the community more or less converge into only a few phyla left in the acclimatized sample. And what's left is mostly from the firmicutes phyla, and we know that they are mostly Clostridia, the anaerobic species that is known to be able to produce hydrogen efficiently. And after that, we did optimization of the experiment based on several physicochemical parameters, such as temperature, what is the optimum temperature for hydrogen production, what is the initial pH, and also what is the inoculum concentration. So the optimization was based on both hydrogen production, and on this side is for COD removal efficiency, because we want both output to be optimized so that we can do maximum waste treatment based on COD removal efficiency, at the same time generate hydrogen at the optimal level simultaneously. So from the design of experiment, software optimization, and then we replicate the results in the lab, we found that there is about 20 to 30% increase in production of hydrogen compared to non-optimized condition. So this was done at a laboratory scale, which is about 60 milliliter scale, and then we scale up the process to 2.5 liter bioreactor, and the 2.5 liter bioreactor, we monitored further hydrogen production, hydrogen content in the biogas, and also changes in pH. So in this figure here, we monitor the different volatile fatty acid production. This is just to know what is the pathway being taken by the microbial community in the reactor, and also later on, when we want to optimize this reactor further with the knowledge on the VFA production, we can actually tweak the process to favor certain VFA so that the biohydrogen production is also optimal. And so we have basically completed the first half of the project, and currently my PhD student is working on the bioelectrochemical system, and we have actually almost completed the testing of the MFC and MEC, and we are now characterizing what are the community of microbes in there, and also try to observe biofilm formation at the electrode. And hopefully soon, if it's possible, we can try to see how is the performance when we integrate both fermentation and bioelectrochemical system in one system. And if you remember this figure just now, I showed that we exposed the microbial community in the anaerobic sludge to increasing concentration of POME. So what we would like to know now is we know the community changes during the acclimatization, and so far we do not know if there is any effects on the functions of the community in the anaerobic sludge. So that's what we are doing now, mostly by my MSc student, Eka. So we will do pre-treatment of the anaerobic sludge, and also after that expose it to increasing concentration of POME, like what we did before, and collect sample at different time interval. And then we will do a more deeper sequencing this time using shotgun metagenomics in order to look at the whole genome of all the microorganisms in the community and try to predict the functional changes of the community in the sample using sequence analysis. So that is the two projects that we are actually working on at the moment in the lab. Okay, so now moving into my research at NUS. So at NUS, I was, the theme is still around microorganisms for environmental application, but the focus is now shifted towards agricultural applications. Because in Singapore, for your information, they have this concept called 30 by 30 vision, which is to secure at least 30% of their food supply locally by 2030. Currently, I think the amount of food that they source locally is less than 10%. And when something like the pandemic happens, border is closed, so they found that it can pose a challenge for them, for their food supply. So there is a very active initiative by the government, by the Singapore government, to do extensive research locally so that they can develop their own food source from different sources, whether agricultural, aquaculture, and so on, so that they can get at least 30% of their food supply sourced locally by 2030. And my group, led by Professor Sanjay Swarup in the Metabolites Biology Lab, is actually focusing on phytobiomes, which is a microbiome or microorganisms that is associated with plants. So if you're not familiar with phytobiomes, this diagram here nicely summarizes the different component of plants, for example, leaf, rhizosphere, which is the root area, and then we also have root endosphere inside the root. Rhizosphere is outside the root, and all these component of plants in the environment is actually influenced by many different types of microbes. So whether it comes from the atmosphere that will colonize the leaf area, and from the soil that will colonize the rhizosphere area. So to research the whole thing is actually very difficult. So in our group, we divide our research team according to leaf area and also root area. And I was tasked to investigate further microbiomes in the root area. So the numbers here represent how many cells are there in the different root compartment. For example, in the rhizosphere, we have about 10 to the power of 6 to 10 to the power of 9 microorganism cells per gram of tissue or of soil. And now we know that these different components are so much affected by the microbes. And in order for us to optimize plant growth, we can actually manipulate the community in the root and also in the leaf area, so that they can work together with plant to optimize plant growth. And this is the strategy that is being taken by our laboratory by looking at the different plant compartments to assess the microbiome presence in there, and also different plant species, different growth stages, different growth condition, and so on. And from the microbiome that we collected, we sequence it using omics, metagenomics method, and then try to map the network and predict the function, if there is any interaction between the individual taxa in the microbiome of a plant. And then from omics method that is non-culturable, we are actually interested to isolate as many as possible individual strains from the different plant compartment, so that we can engineer the isolated strain using the desired consortia that we want, and test this consortia on the actual plant in the greenhouse, so that we can improve plant productivity based on the nutrition, based on plant defense against pathogen or biotic factor, and also to improve the growth. So my focus was to look into leafy vegetables, or we call it leafy Asian greens. So Singapore, I think, is a very big consumer of leafy greens. So researching into leafy greens, vegetables is actually very attractive to the Singapore government and Singapore society. And the first, I think, important paper that the group published was just last year. So from the microbiome that we collected from the different plant compartment, we found that we could get up to 900 metagenome assembled genome, meaning these are the number of microorganisms present in the different plant compartment that can be sequenced, and we can even actually assemble the sequence to get the whole genome. And this whole genome is very useful because we can then know who are they, and then what they can do in the environment. So this will guide us to select the desired strain that we want, and then try to construct synthetic consortia that we can reintroduce to the plant to improve the growth. So leafy green is actually also widely used in Singapore's urban farms. So they have been planting leafy greens on rooftops, and also in different, because they don't have much land. So urban farm is also very popular in Singapore, and leafy greens are among their choice of crop, their main choice of plant as food source. So our strategy is to use our self-developed system called Live Exudation-assisted Phytobiome. So you can see here, this is just like a normal petri dish, but it's square dish for plant tissue culture. So what we did was to grow microbiome on the bottom level of this plate, which is on agar, and then we put membrane on top to separate plant from the microbe, and then we grow seedlings on top of the microbiome. So the choice of plant for me is Brassica rapa, or in our, we are more famously known as choisum. I think some of you know what choisum is. So the idea is to separate the seedlings from the microbiome at the bottom with this one layer of membrane, so that there is only chemical exchange happening between the microbiome and the seedlings. So our idea is to avoid colonization of the seedling by the microbiome, at the same time to find out what are the chemical that is being used by the microbiome or the seedlings to communicate with each other. And from this simple system, we can actually get a lot of information, whether from the seedlings, which is what are the exudation by the root using metabolomics. And then we can also look at the membrane, where we can find what are the exudates that is being transferred from seedlings to microbiome. And from the microbiome, we can know the different changes of the community affected by exudation from the seedlings. So it was mainly developed by a PhD student in the lab, Yong Liang, and also postdoc, Dr. Saxena. So the overall strategy is to actually harvest inoculum. So in this case, choose some grown in pot and also control potting mix with no plant. We harvest these two and then use it as an inoculum to inoculate on a square plate in a LEAP system like I've shown earlier. And then after a while, when the microbiome has been exposed to plant influence, then our intention was to harvest the cells from the agar layer and then stain them using, for example, live dead staining or any specific probe that we want, if we want to target specific texture, and then put it through flow cytometry or FACS, fluorescent activated cell sorting, to sort individual cells. So it can be soft sorting to allow coexisting cells to be sorted together, or hard sorting, with which will sort all the single cells. And our intention was to take this sorted cells and then grow it in a high throughput culturing system in order for us to try to culture as much or as many as possible strains from this LEAP system, which is representative to the rhizosphere of plants growing in the soil. Okay, and then from the high throughput culturing system, we can actually get a lot of information from it that can further guide us to develop consortia and can be introduced back to the soil in order to improve plant growth. So this method actually avoid the use of chemical fertilizer. So that is the ultimate goal of the research, which is to avoid use of chemical fertilizer, and it's only based on the engineering of microbiome in the rhizosphere of plant to help it produce a healthy plant. So before this, we found that there is a difference in so the LEAP system can actually influence microbiome on the plate, as you can see here. From day zero, this is the absolute abundance of the microbes in the system. On day zero, this is the system inoculated with root microbes or rhizosphere microbes, and this is the system inoculated with soil microbes. And after 7 days, we can see that there is an increase in abundance of the microbiome inoculated with root microbe compared to soil microbe, which is used as a control. So this is at the phylum and at the genus level. But the problem at that time was we could not get the phenotype of the plant, meaning the system could not improve plant growth, even though we can see some changes in the microbial community on the plate, but the enhancement of plant growth could not be observed at that time. So what I did was to first quantify how many microbial cells are present in the bulk soil, or which is used as a control, and also rhizosphere or root environment. And I found that the rhizosphere environment has higher cell count, which is expected about 10 times more than bulk soil microbiome. So this is a useful information when I was doing the LEAP experiment in order to equalize the number of cells used in the inoculum. So after a lot of optimization using different media for the microbiome, different light density, and also different number of seedlings on LEAP plate. So I managed to come up with an optimized system for use in the LEAP, You can see here the different growth stages of choice-sum seedlings on plate with no microbiome, on bulk soil microbiome, and rhizosphere microbiome. It is still hard to see enhancement of the plant by rhizosphere microbiome or bulk soil microbiome based on the different data collected during the investigation. So what I did was, from this microbiome growing on the LEAP plate, I reculture the root influence microbiome onto a second round of LEAP plate. So we could see that using the re-inoculated LEAP microbiome in the early stages of growth, especially using bulk soil microbiome, I could see enhancement of root hair in the conditioned microbiome compared to heat-treated microbiome, which is used as control, and also no microbiome. So we can see that there is enhancements in root hair formation, and this enhancement in root hair formation is actually accompanied by a higher plant weight in the early growth of the seedlings. So this does not happen when we incubate it further. So it is another interesting observation that we are now investigating, what actually contributes to the enhancement in root hair formation here, whether there is an active interaction between the conditioned microbiome and plant, and what are the chemicals used in the interaction to lead to this enhancement. So there is an ongoing research for this project. So we have completed the first part of the project, and if everything goes well, we will then proceed with the flow cytometry or flow sorting of the cells from the LEAP system, and then proceed with the high throughput cultivation. So at the moment, we have also secured two big projects from Petronas. The first one is from Petronas, which will investigate the methanogenic conversion of carbon dioxide to methane for CO2 storage. So for this project, we will be using archaea, known as methanogen, to try to convert CO2 into methane, because if we can convert CO2 into methane, we can actually use it for CO2 storage and also generate new source of fuel in the form of methane, if everything goes well. And at the same time, we have also just secured another grant from A*STAR Singapore to investigate the conversion of methane to single cell protein. So this is another research under the Singapore 30 by 30 initiatives, because if we can convert methane produced, for example, from anaerobic digester, we can convert it to single cell protein. Then this single cell protein can actually be used as animal feed that will support aquaculture industry in Singapore. So this project is actually at the early stage, and this A*STAR project has not started yet. We are slowly ramping up this project by currently recruiting postdoctoral candidate, and hopefully in June or July or August, the candidate can join, and then we can start our project on this grant. So I hope you have you managed to appreciate the sharing, because I started with all the way from my MSc, and how those experiences from MSc, PhD, and the early stages of my research career shaped the research that I'm doing now. And I think, as I mentioned earlier, my interest is slowly being shaped up, and I know what I would like to do now, and hopefully more and more grants can come in to allow us to investigate the potential of microbiomes or microbes to enhance different processes, whether environmental, agricultural, or industrial application. Thank you for your attention.
Okay, thank you, Doctor, for your impressive presentations. Now we open for Q&A sessions.
Assalamualaikum.
Waalaikumsalam.
My name is Sohana. I have a question to ask. Thank you, Doctor, for the nice presentation. My question is, in terms of cost involved in turning the microorganism for the environmental and industrial application, is it more cheaper compared to the normal or current treatment? And if it is not cheap, how do you tackle that in order to be able to be implemented in the industry?
Okay, thank you, Sohana, for the question. So the idea of using biological method is, as you said, is the cost effectiveness, because when we use microbes, especially if we can source them from the environment, it is very cheap. There is very minimal cost involved, and for me, the main cost for biological processes is not sourcing the microbe. It's actually in the optimizing the process, because getting the microbe is easy, but getting them to do what we want in the process is not. So I think more investment is needed in terms of finding the right condition, finding the right media that they can grow in, that they are happy in, so that we can get them to do what we want. So yeah, getting them is not costly, but maintaining and forcing them to do what we want requires more money there. Hope I have answered your question.
Yes, Doctor.
Doctor, I would like to ask. Hello.
Sure. Yeah. May I know who's speaking?
I'm Azhar. For degradation of POME, and we produce biohydrogen, and then I would like to know, is it are you going to do in a large scale? That means you produce biohydrogen in large scale for renewable energy. That means replace the current electricity or somehow like that.
Yeah. Actually, that's the idea, and if you read more about research on biohydrogen from POME in Malaysia at the moment, it's not just us doing this research. There are a few other groups that actually has gone through to pilot scale. So if you remember what I was presenting, our research is still at lab scale, but the difference between us and others is the temperature used, because from the studies done, they found that the optimum temperature for hydrogen production is actually quite, I would say, relatively higher. So in my group, we do not want to go that high, more than 40 degrees, for example, because if we want to run the process at higher temperature, you will need energy. So our research is now focusing on mesophilic temperature, we call it, from 30 to 40 degrees. So at mesophilic temperature, if everything goes well, we can run the process at room temperature only. And yes, the idea is, of course, to bring it to pilot scale, like what others are doing. But before we go to larger scale, we have to understand fully the process at this lab scale first, especially the biochemistry, the microbial community behavior, and of course, with support from different agencies. Hopefully we can bring the process to higher scale, and the ultimate aim is of course to introduce the system to the industry, for them to use it to generate electricity, at least can supply the requirement of the mill, if not to give back to TMB if the production is high. Yeah, thanks for the question.
Good morning, Doctor.
Good morning.
Thank you for your interesting presentation. So I would like to ask one question just now, that regarding the future project.
May I know who's talking?
I'm here.
I think I know your voice.
All right. Yeah. Thank you. So regarding the future project that you work with A-Star Singapore, is about the single cell protein. So is this is it a conversion of the methane to single cell protein? So what is single cell protein?
Yeah, thanks Heo for the question.
Thank you. Yeah.
So single cell protein here is defined as a microbial cell. So when you grow the microbes in the presence of the substrate here is methane. So you pump methane into the reactor, and this microbe can use methane to convert it into their cell, which is to grow. So we call that single cell protein.
Right. And this single cell protein will be used as feed, like animal feed or something like that?
Correct. So before we can use it as animal feed, of course, there is a prelim step required to purify the single cell protein after we have grown them in a reactor. So we remove all the other unwanted media, leaving only the single cell. And we need to do nutritional analysis to see if it suits for animal feed applications, especially in terms of protein and nucleic acid content. And if everything is okay, then we can test it for use as an animal feed.
Yes. Because I was thinking about the sustainability, food waste. For example, food waste, decomposed food waste. Just talking about the landfill. The landfill, we have a lot of food waste pile up, and they at the end they will produce CO2 as well as methane. So I was thinking, like, is it possible if we can accumulate those methane produced by the landfill, and then through the help of the microbes, it can convert into, like what you say, the single cell proteins, which is potentially can be used as an animal feed. So it's a perfect circular economy. What do you think?
Yeah. That's a good suggestion, especially for Malaysian application, because in our case we have landfills, but in Singapore they do have landfills, but they don't use it for methane production, meaning they do not tap methane that is coming up from there.
Yes.
If I'm not mistaken, they bring all their trash to this
incinerator.
It's not incinerator, but this man-made island. So they have this Macau island where they use the waste to slowly, reclaim the sea, and use it as nature reserve. So it's very impressive what they're doing.
Correct.
But they also have this anaerobic digester for domestic waste. So that's what we propose. Where the methane can be sourced in Singapore's context. But if the technology works, it can actually be used in any methane producing facility, like what you said, landfill and so on.
Yeah, because I've seen our own landfill in Selangor. They actually produce methane and also to generate electricity. But this could be another potential application where, if we can grow proteins out of the methane. Yeah, it's
and
a great idea.
avoid CO2 production further.
Exactly. Yeah, inspiring. Thank you so much, Doctor.
No problem. Thanks for your question and idea.
I think if there's no more question, we can close this session now. So, Dr. Firdaus, anything you want to say before we wrap up?
I just want to say thank you to all of you for joining the session. I know my talk is not actually medically relevant because I know all of you are probably from medical field. But the techniques that I was talking about, for example, the omics, metagenomics, and autometabolomics, can actually be used in medical applications too. And probably, I'm sure some of you are already working on that. Yeah. Thank you all, and I hope to see you all again in person.
Okay, thank you, Doctor. On behalf of IMB, I would like to thanks to Doctor and all of you that joining this webinar. Till we meet again. Bye.
Thank you, Dr. Firdaus. Thank you all for coming.
Thank you all.
Thank you.